Hello and welcome to this webinar titled A New Paradigm of Building Quality During Manufacture, Challenges with Biologics. Hosted by Biopharma Asia Magazine and presented by Dr. Rajesh Gupta from Biologics Quality and Regulatory Consultants. Following Dr. Gupta's presentation, there will be a follow-up presentation titled Considerations for Manufacturing Commercial Antibody-Drug Conjugates, a CMO Perspective. This will be presented by Dr. Joji Swami and Josh Morris from Millipore Sigma. Also from Millipore Sigma, we are joined by Mark Cooley. Mark will help answer any questions you may have at the end of the webinar. My name is Afsal Mir and I will be the moderator. Before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce you to our first presenter. Dr. Rajesh Gupta has a PhD in microbiology and is a principal consultant at Biologics Quality and Regulatory Consultants, LLC. He has more than 35 years experience in the development, production, testing and regulation of biologics, working at both the regulatory agencies and within the industry. Whilst at the FDA at the Center for Biologics Evaluation and Research, he was a Deputy Director and Lab Chief in the Division of Biological Standards and Quality Control, managing lot release of biological products, regulatory reviews of analytical methods in the biologics license applications, and generation of reference standards and development of new methods. In his previous jobs, Dr. Gupta worked at the Biologics Consulting Group in Wyeth, Chiron, Massachusetts, at Public Health Labs, and at the National Institute of Health in the USA, as well as the National Institute of Immunology and Central Research Institute in India. His major accomplishments are in adjuvant and delivery systems for vaccines, polysaccharide protein conjugate vaccines, combination vaccines, and development and in the validation of analytical methods. In these areas, he has published more than 100 papers in peer-reviewed journals and books and made numerous presentations at scientific and regulatory meetings. Welcome, Dr. Gupta.
Thank you, Afzal. Greetings to everyone around the world, and today I'm going to present on the paradigm of building quality during manufacture for biological products. As most of you may know that this is a huge topic, particularly for biological products, and therefore this webinar presents a high-level overview, particularly on the challenges in building quality in biological products, with the limitations of not discussing the topic in details due to time constraints. And in this presentation, I will start with going over briefly on the traditional paradigm of building quality through final testing of representative samples, then moving to the current paradigm to build quality in the products during manufacture, specifically listing challenges in implementing the quality by design concept for biological products. Then I will go into ways to build quality in the product, discussing briefly implementation of GMP regulations to complex biological products, quality by design concept, brief introduction of production analytical technologies, role of life cycle management of product, FDA's new initiative on quality metrics, and finally the benefits of building quality in the product. Now, starting with a brief introduction about the biological products, as you all know that these are important class of drugs used in diagnosis, prevention, and treatment of disease. Often, these biological products are at a cutting edge in medical science and research and first in class, posing challenges in determining regulatory pathways based on established laws and regulations. US FDA regulates biological products based on science and law and often issues guidance documents in regulation of new biological products or implementing drug laws on the existing and new biologics. For new innovative complex biological products, it is always recommended to have consultation with the FDA, or I would say even any other regulatory agencies in Europe, to determine the regulatory pathway, because based on science, benefits and potential risk of the product, the regulatory pathway may be decided case by case. Certain biological products such as vaccines are given to healthy population with almost no or very low acceptable risk, which makes ensuring quality or building quality in the product a critical component of the manufacture. And often final bulk or final products cannot be tested effectively for safety or risk due to adventitious agents, particularly adventitious viruses. Thus, quality must be built into the product. Conventional quality paradigm for drugs and biologics has been to ensure quality by testing representative samples. Historically, biological products, mainly vaccines and antitoxins, which I call legacy biological products, were made in a relatively flexible manufacturing environment with rigorous testing throughout manufacture, including testing of critical raw materials, intermediate products, final products, and final product. Most legacy biological products, vaccines and antitoxins, were developed empirically with manufacturing processes based on experience and science, and were not prospectively validated for consistency in manufacture. Though there was a strong belief that the process is the product for the legacy biological products, the manufacturing process was not strictly controlled and well documented, particularly with regard to changes and deviations to the routine manufacturing process. The major focus to ensure quality in these legacy biological products was on testing, often in animals for safety and potency, and releasing the product based on results from representative samples. Product monographs describing testing and specifications for most of these products at various stages of manufacture were included in the regulations. For example, some of you may be familiar with 21 CFR Part 620, Part 630 and pharmacopoeias. Even European Pharmacopoeia gives these monographs even today. But these 21 CFR Part 620 and 630 were deleted almost 10-15 years back. So during 1980s, recombinant biotechnology brought a number of modern biotechnological products in the market, mainly for therapeutic applications, which comprises highly purified recombinant proteins or other biological products. These products were developed and manufactured in new and modern manufacturing facilities following Cgmp regulations. During 1990s then, regulatory agencies in the United States, Europe and the World Health Organization started implementing Cgmp regulations to the manufacturers of legacy vaccines, immunoglobulins and antitoxins, requiring these manufacturers to validate facilities, manufacturing processes, other systems and analytical methods using retrospective data. In addition to ensuring quality of biological products by testing representative samples throughout the manufacturing process, implementation of Cgmp regulations included following the concepts of consistency in manufacture through the quality management systems, documentation systems, validation of facilities, processes, utilities, methods and cleaning procedures, increased systematic and documented monitoring of facilities, environment, utilities and processes, using change control procedures, following through deviations, doing thorough investigation, etc. So they were following all these Cgmp related these principles or quality management systems. They were still doing representative testing on the product. So with the view to providing flexibility in implementing new technology and developments for the legacy biological products, a number of product monographs specifying testing requirements were deleted from regulations. For example, as I already mentioned, in the US, 21 CFR parts 620 and 630 were completely deleted. And many vaccine monographs were deleted from some pharmacopoeias, particularly the United States pharmacopoeia. So since the beginning of 21st century, the FDA has taken major initiatives in regulation of drugs, including biologics, using risk-based and science-based approaches, providing a new paradigm that quality cannot be adequately assured merely by in-process and finished product and inspection or testing. Quality must be built into the product during development and manufacture. Then they came up with this terminology, quality by design, which involves use of scientific quality risk management principles and quality control strategies based on understanding and knowledge of the product and the process. So to build quality in the product, it is essential to gather and understand scientific and technical knowledge about your product and the manufacturing process during product development. This includes identifying critical quality attributes, critical starting materials, and critical raw materials such as seed stock, cell banks, growth media, and defining critical process parameters affecting quality. During process development, relationships between these critical raw materials, critical process parameters, and critical quality attributes is also evaluated and determined if possible. Because as I mentioned that the biological products are very complex and some of these may not be possible to determine exactly the relationships between these parameters. The manufacturing process then will include online or at-line monitoring of these critical process parameters and critical quality attributes with the ability to adjust the critical process parameters within the design space. So due to the complexity of both biological products and the manufacturing process and unique challenges with regard to both drug substance and drug product manufacturing, there are challenges in applying the QBD principles to the biological products. Therefore, CMC working groups from the industry with representatives from major biotechnology and pharmaceutical companies, and also there were representatives from the FDA were present. These CMC working groups were formed to provide some examples of application of QBD to a hypothetical monoclonal antibody molecule and a vaccine products. These documents are available online. So some of the challenges in applying QBD to biological products include requirement for stability of biologics in design and control of the manufacturing process, and then understanding and managing inherent variability in manufacture, which is due to the fact that starting and raw materials for manufacture of biologics are of biological origin. Biologic processes are complex, intermediates are unstable, and sometimes they support microbial growth. And finally, the use of bioassays in the manufacture and release of these products, which have large inherent variability. So then how to deal with the challenges? That my experience has been that you should focus on science and in understanding the product and the process and the variability as much as possible. So once you understand the process and product and identify the variability as much as possible, then you can develop plans to manage the sources of variability. So I will just assuring microbiological quality of biologics. As already known, microbiological quality must be built into the product by understanding the sources of contamination, environmental conditions, and product attributes that support growth of microbes. Because microbiological methods and testing do not provide assurance on the complete absence of viable organisms. Biological products cannot be terminally sterilized. Therefore, microbiological quality is assured by employing a robust environmental monitoring program, appropriate microbiological testing at various stages or intermediate products during manufacture, including the final drug product, and using validated manufacturing processes. For example, aseptic manufacturing processes, container closure studies, media fill studies, etc. are done to validate these processes. And then validation for reduction, removal, and inactivation of potential microbial contaminants, including viruses, mycoplasma, and bacteria, must be performed for the selected manufacturing processes that have the potential for reducing the microbial bioburden. So building microbiological quality during manufacture of legacy biological products has been a key factor in the success of these products, despite not following Cgmp regulations during manufacturing operations until 1990s. So the message is that you need to build microbiological quality, which has been being done for most of the legacy biological products. So now I will comment a little bit on the bioassays because that's an important topic for the biological products. Bioassays or biological products are commonly used to evaluate functional effectiveness and safety of biological products. So as per the name, bioassays use a biological system in the test method, including tissues, cells, animals, and microorganisms, so that itself introduces additional variability despite the complexity of your test material. So bioassays have been used for potency and toxicity testing of many vaccines and other biological products also. So these could be quantitative assays based on absolute quantitation or performed using reference standard. So a few examples are given in this slide. Then the bioassays can also be quantal type of methods, which are often used to determine 50% endpoints such as LD50, ED50, or potency of viral vaccines like TCID50. And again, some examples are given here. And finally, there are bioassays that are limit tests, and a few examples are given in these slides, which I am not going to go over in detail. And just to enumerate a few challenges with the bioassays, that as mentioned, that due to complexity and diversity of biological products and use of biological system in bioassays, these methods have low precision as compared to other analytical methods used in testing of drugs. In fact, it is a common myth in the biologics industry to accept validation of bioassays with a large variability having 25% or higher coefficient of variation for precision. So in my experience, any analytical method, including bioassays and microbiological methods, with a CV of 25% or higher will not be appropriate as a quantitative method that could support product specifications as wide as 50 to 150%. So therefore, selection and design of a bioassay with acceptable accuracy and precision supporting intended use of the method and validation of such method require in-depth knowledge and understanding of science, quality, and regulatory aspects about the product. The second point on bioassays are that since biological products are often cutting edge in medical science and research and first in class, as I already mentioned, test methods and standards are not available. These create challenges in demonstration of specificity and accuracy for these methods. But there are challenges, but despite these challenges in developing and validating accurate and precise bioassays, if you know the product, know what do you want, so you can develop successfully bioassays which have been used, I would say, more than a century in manufacture and testing of biological products, particularly vaccines, immunoglobulins, and antitoxins. So the message is understanding science for the bioassay and focus on intended purpose of the method are important in development of precise and accurate bioassays. So coming back to the quality by design concept, which is a science and risk-based systematic approach to product development starting with predefined objectives. Main focus of QBD is to understand the product and the process based on sound science and quality risk management principles. All attributes, which includes quality, product and component, and parameters, which include process, operating and equipment, should be evaluated for their roles in the process and impact on the product or in-process materials, for example, intermediate products. Based on the understanding and knowledge of the product, the process, and relationship between these critical raw materials, critical process parameters, critical quality attributes, then design space is defined for the critical process parameters. Finally, the process is designed with online monitoring, having ability to adjust the critical process parameters, of course, within the design space by measurement of critical quality parameters. So this will lead to continuous real-time quality assurance. Then coming to the process analytical technologies, or PAT, which represent tools or a system to design, analyze, and control process to implement QBD by continually monitoring and updating process to assure consistent quality. For an effective and successful implementation of this PAT, it is essential to understand scientific and engineering principles of manufacturing process and equipment and to identify variables affecting quality. PAT is able to monitor and control process in real time with feed forward and feedback controls. Information collected through PAT is used for continual improvement by tracking and trending of process operations. So this PAT initiative, in fact, FDA started this discussion publicly in 2001 and coming up with the guidance in 2004. And FDA also came up with the Pharmaceutical Quality for 21st Century initiative, which include both the fundamental concepts of QBD and PAT. So now a couple of slides on the analytical methods, because the process knowledge depends on accurate and precise measuring techniques used to test and examine the quality of drug components in process materials and finished products. Selection of appropriate analytical methods and online tools for PAT and for identifying and defining CQAs, CRMs and CPPs is extremely important. Recent advances in technology in analytical sciences and instruments and developments in proteomics and genomics have provided powerful tools to characterize biological products. Analytical methods based on these technologies are highly sensitive, precise and accurate in characterizing biological products, specifically in assessing their purity. These methods, along with the concept of well-characterized biologicals, are extensively used in applying principles of QBD to modern biological products. However, there are challenges in implementing these technologies to the legacy biological products. So, since the QBD process starts an early product development, methods used to identify and define these CQAs, CPPs and CRMs will not be fully qualified and validated. And this question has been asked to me often when even I was at FDA. So at that time we had some discussion and came up with that these methods should be at least as reliable as characterization tests, which are used extensively during product development to understand the product, for example, structure and purity of the product and mechanisms of actions, proof of concept, but are not part of release tests. These tests should be scientifically sound, for example, specific, sensitive and accurate with robust method design, and should be suitable for intended purpose for which you are using. So during commercial manufacturing, characterization tests are used in support of changes to manufacturing process or other changes to approved license applications. When analytical probes or chips are used for PAT purposes, these should be appropriately calibrated and or verified at periodic intervals. So now, after implementing these QBD, what are the benefits of this new quality paradigm? The manufacturing process developed on the QBD principles will be robust, based on science with flexibility to make changes within the design space without regulatory notifications and approvals. Such a manufacturing process can be continually improved for enhanced compliance and efficiency and a better product. With upstream quality control using PAT, there is a possibility of real-time release or reduced end product testing, resulting into reduction in cycle times. Robust manufacturing process will provide a higher level of assurance of product quality with reduction in product reject and waste. The manufacturing process will be amenable to accommodate new scientific knowledge and modern technologies with increased use of automation and new methods, resulting into improvement in the product. So one of the concepts in this whole QBD is the life cycle management of the product. So that is achieved by monitoring and managing the process. So as you know that historically even manufacturing process for legacy biological products, though not much controlled and documented as discussed earlier, has been randomly monitored as part of the belief that process is the product. Example of this process monitoring for legacy products including like monitoring of pH, dissolved oxygen levels, glucose levels during fermentations, and also some offline microscopy examinations like gram staining as indicator of purity for some of the specific vaccines by immunochemical tests like flocculation test, agglutination test as indicator of yield, and tests of intermediate products like lethal toxins, lethality, indicator of quality of the product. So that's what I wanted to emphasize here, that legacy products had certain elements of building quality, and that's why we had very successful products like eradicating smallpox, the polio vaccines, or some of the legacy vaccines, antitoxins or immune globulins have been very, very successful. And based on these, then in the past also the actions were taken to terminate or continue the process. So continuing with the process monitoring as a part of the life cycle management, in the FDA guidance on process validation, the focus is to develop a manufacturing process that makes drug fit for its intended use, based on the philosophy that quality, safety and efficacy is designed or built into the product, and it cannot be adequately assured merely by in-process and finished product testing. Each step of manufacturing process must be controlled to assure that finished product meets all quality attributes including specifications. So each step of the manufacturing process is controlled. Then even in the Cgmp regulations, they require that manufacturing processes be designed and controlled to assure that in-process materials and finished product meet predefined quality requirement consistently and reliably. So goals of manufacturing process validation and continuous process verification or monitoring are to ensure homogeneity within a batch and consistency between batches. That is what is consistency in manufacture is. So as mentioned already, the attributes are quality, product, component or critical raw materials, and the parameters are process, operating and equipment. These may be re-evaluated as more information is available from continuous verification of the manufacturing process for their roles in the process and impact on the product or in-process material. Degree of control over these attributes or parameters should be as per their risk to process and process output. So the message is that you have to manage the life cycle of the product, and including actually analytical methods also. It is not that once you are done with the validation, either process validation or method validation, the job is done. You have to continually monitor and track and trend these processes or methods so that you manage the life cycle, and if any changes are to be made, these should be made to keep these methods in a validated state so that they are suitable for intended purpose. So recently in July 2015, FDA issued a draft guidance on request for quality metrics in order to further their goal of a maximally efficient, agile, flexible pharmaceutical manufacturing sector that reliably produces high quality drugs without extensive regulatory oversight. So the intent was very good, that they want to reduce the regulatory oversight on the drug manufacturers. Goals of requesting quality metrics from drug manufacturers included promoting responsible practices and quality-driven corporate culture, identifying situation for a risk for drug supply disruption, and improving FDA's evaluation of drug manufacturing and control operations. So quality metrics are an objective measure of the quality of a product or process, an objective measure of the quality of a site, an objective measure of the effectiveness of systems associated with the manufacture of pharmaceutical products, including the pharmaceutical quality system, and required even under Cgmp, like you know that annual product review manufacturing includes some of those data, statistical process control charts, and process capability outputs. Based on comments from the industry, FDA issued revised draft guidance in November 2016 on submission of quality metrics data with significant changes from the original draft guidance. In the revised guidance, FDA is requesting data on 3 quality metrics: lot acceptance rate, product quality complaint rate, invalidated out of specification rate. So initially there were more parameters they were considering, or metrics they were considering. So though FDA revised this guidance, but months after FDA revised its draft guidance in response to industry's concern, a consortium of industry groups, including major pharma and biotech manufacturer associations, are asking FDA to halt its plan to move the program forward. So we don't know the future of this yet. So now the last slide, just quickly on the continuous process verification, provides that the manufacturing process remains in a state of control. The validated state during commercial manufacture is a critical component of assuring quality of the product throughout its life cycle. So the continuous process verification is done by monitoring of data, observations, and results for compliance with standards, specifications, or limits. So there are 2 types of data I have categorized: data of compliance that meets standards, specification, or limits, which is used to release product. Such data are tracked, evaluated for trends with periodic reviews. Data of exception do not meet standards, specification, or limits, and include deviations, non-conformances, out of specification results, and invalid results. Such data and observation need immediate attention, notification, and investigations. So finally, to conclude that historically, the quality of biological products has been ensured through testing representative samples. Shift in quality paradigm started with implementation of Cgmp regulations for legacy biological products in 1990s. In 21st century, quality paradigm for drugs shifted to building quality during development and manufacture rather than testing alone. Critical aspects of quality by design philosophy include focus on science in understanding product, process, and variability, and designing processes on sound scientific principles and quality risk management principles. Inherent variability and complexity of biological products pose challenges in implementing QVD concept. Analytical method must be scientifically sound and reliable, as quality from the manufacturing process depends upon these methods. And then, as I mentioned, life cycle management of product ensures that the quality of the product remains in the validated stage. Regulatory agencies support and encourage use of QVD approach for new products and has provided valuable information in a number of guidance documents, which is given in the next slide, some of these documents. While these initiatives and efforts by the regulatory agencies and manufacturers are commendable to ensure quality of drugs, it is disheartening and scary to see some manufacturers compromising quality by manipulating and falsifying data and interfering with regulatory inspections. Ensuring quality of drugs is a joint effort by regulatory agencies and pharmaceutical manufacturers. Manufacturers have a major role in ensuring quality by building it in the product and working in cooperation with regulatory agencies to assure the public that when they take a medicine, there should not be any worries or concern about its expected quality. So as I mentioned that there are a few references taken, and these references to those 2 examples of the CMC biotech groups, which they gave, like how to apply QVD on a hypothetical monoclonal antibody or vaccine, are in these references. With that, I conclude my presentation. I thank you everybody for attending today's webinar, and now pass on this mic to Ajfal.
Thank you, Dr. Gupta. Just another reminder to our audience that if you have any questions, please click on the questions tab at the bottom of the screen and send them to us during the webinar, and we will go through these at the end of the presentation. In order to answer any of these questions, we are joined by Mark Cooley from Millipore Sigma, who has 21 years of experience in the pharmaceutical industry and has held a variety of management roles in quality control and quality assurance. Mark has been with Millipore Sigma, a business of Merck, in Darmstadt, Germany for 7 years and is currently the head of site quality for the St. Louis Cherokee site, which manufactures APIs, excipients, and biological buffers for the pharma industry, including antibody-drug conjugates. He received bachelor degrees in chemistry and biology from the University of Northern Iowa. Now begins the second half of this webinar, and I will now introduce our second speakers who will be doing a follow-up presentation on considerations for manufacturing commercial antibody-drug conjugates, a CMO perspective. Our first presenter, Dr. Joji Swami, has been in the pharmaceutical industry for over 25 years, covering areas ranging from analytical method development, method validation, cleaning validation, stability, project management, and marketing. Joji has been with Millipore Sigma, a business of Merck in Darmstadt, Germany for over 11 years in various roles including senior chemist, quality control supervisor, and senior project manager. Joji is currently the head of global marketing focusing on ADCs and was an integral part of the Millipore Sigma and Baxter collaboration, working out the integration process with Baxter's counterparts. She received her Bachelor of Science at Bangalore University in India and her Master of Science at Indian Institute of Science in India, along with her PhD in Synthetic Organic Chemistry at the University of Missouri in St. Louis. Our second presenter, Josh Morris, has been in the pharmaceutical and biotechnology industry for over 7 years in positions including project management, GMP biologic contract manufacturing, process analytical development, and quality control. Josh has been with Millipore Sigma, a business of Merck in Darmstadt, Germany for 4 years in roles including senior manufacturing scientist and currently as a custom manufacturing project manager. As both an individual contributor and a project manager, Josh has led commercial biologics readiness operations supporting the Millipore Sigma antibody-drug conjugate commercial manufacturing facility. Josh has received a Bachelor of Science in Chemistry from Old Dominion University and a Master of Science in Chemistry from Southern Illinois University in Edwardsville. Welcome, Joji and Josh.
Thank you, Afzal. Good morning, good afternoon, good evening. We appreciate everyone calling into this webinar. I will co-present with my colleague Josh Morris in letting you know our CMO perspective on considerations for manufacturing commercial ADCs. Outline of our talk is here. I will briefly talk about history and future of ADCs, and Josh will go into details on CMO considerations for ADC manufacturing. Some of them are listed here: facility design requirements, EM strategy, process development activities, cleaning strategy, regulatory and process validation strategy. At the end, he will summarize our talk. Here is a slide defining ADC. I'm sure everyone knows what ADCs are. Since this talk is about ADCs, I thought I would include a slide. I will really not go into any more details on this. History of ADCs globally. ADCs are fairly new compared to small molecule drugs. Mylotarg from Pfizer was the first ADC that was approved in year 2000. It was later withdrawn from US and EU markets in 2010 due to side effects. However, Japanese market decided to keep the product available for patients based on the benefits versus risk for the drug, from the drug. Based on new clinical data, FDA advisory committee voted in favor of Mylotarg last month. So hopefully it will be available for patients in Europe and U.S. markets soon. Adcetris from Seattle Genetics was approved in 2011. Capsula from Genentech was approved in 2013. The very new approval for Besponsa from Pfizer was approved this year. In fact, EU approval was received last month in July, and FDA approval was just last week. That shows how dynamic this market is. You know, things are changing every day. Where are we with the rest of the programs globally? This slide summarizes information from a Beacon report that was published or presented at World ADC Conference in Berlin earlier this year, and also information that was sent out in August edition this year. When you look at number of molecules in 2016 and as of today in 2017, it doesn't look like there is a big difference. But what we need to understand is this 66 molecules that is shown here are not the same that were available in 2016. Many products have been discontinued for various reasons, and many new ones have been introduced into phase 1 and phase 2 clinical trials. What this table doesn't show is the over 250 preclinical programs that are underway that could get into phase 1 or phase 2 clinical trials any day. That shows you the depth of the product pipeline. Even though it is dynamic, it also tells you that it is very robust. One of the recent alerts from Beacon mentioned that the number of approvals for ADCs is expected to double in the next 4 years. So that tells me that ADCs are going to be gaining prominence in the near future. What else can we expect from ADCs in the future? Looking forward, if you look at clinicaltrials.gov, you'll see over 200 trials registered. How is it possible with less than 100 ADCs? We saw in the previous slide there are about 66 ADCs in the program. That is possible because of multiple indications that are registered and also combination therapy. I do expect the number of combination therapies to increase significantly. And the other aspect to expect for ADC is ADCs made with bispecific maps due to increased efficacy from these bispecific maps. Another important aspect of ADCs is use of ADCs beyond oncology treatment. Recently, we have seen many notices and publications telling us ADC use for other indications, other treatments. I chose to put information from a publication from last year that listed immunological modulation, anti-infection, where antibiotic conjugate was used, cardiovascular diseases, liver metabolic disorders. I've also seen some papers where ADCs, where the drug is fluorescent, is used as a diagnostic tool. So it is really interesting that ADCs are going to be used for more than oncology, and that tells me from a CMO perspective the demand for ADCs is going to go up, and as a CMO we should be ready to address any needs that come up. Our organization has been producing small molecule commercial APIs for many years. The site in St. Louis where ADCs are produced has been producing small molecule commercial APIs since 1999. We opened up a clinical ADC facility in 2008 and commercial facility in 2015. To date, we have produced more than 80 GMP batches that are made up of more than 25 different ADC constructs. That tells you the depth of experience that we have had within ADC space, and the many considerations that went into getting this experience and becoming changing our expertise from a small molecule to becoming experts in ADC manufacturing. My colleague Josh Morris will tell you how we got to where we are with respect to ADCs. With that, I'm going to pass it on to Josh.
Thanks, Jyoti. So there's some important consideration and evaluations that a contract manufacturing organization must perform when manufacturing antibody-drug conjugates. It starts with facility design, and ADCs have unique requirements. A CMO must consider the individual components of an ADC, the high-potent small molecule, and the biologic mAb. The challenge here is the requirements for these individual components oftentimes work in conflict from a facility design perspective. Specifically, high-potent small molecules require containment from a safety perspective, and biologics require environmental controls to satisfy GMP needs. Let's talk about some of the facility design considerations, beginning with HVAC. Not unique to ADC manufacturing, you would expect a facility to have single-pass, HEPA-filtered air. More unique to ADC manufacturing, for a containment perspective, you would expect the exhaust from that facility to be HEPA-filtered as well. Additionally, there are some unique air pressurization strategies from within the facility. For a typical biologic facility, you would expect the pressure within the facility to have a high positive pressure compared to the outside of the facility. In practice, what this means is that air from the inside of the facility is pushed to the outside of the facility, preventing the contaminated air, quote, from the outside of the facility from entering the facility. ADC manufacturing utilizes a similar technique, but must also consider the containment aspects for the small molecule. I want to draw your attention to the image to the right, and we're actually going to begin at the very bottom right-hand corner at the exit airlock. You see this exit airlock, which is connected to the outside of the facility, has 3+ marks. This indicates that airlock has a very high positive pressure compared to the outside facility. This prevents the outside air from entering the facility. Now let's talk about the containment aspects. I want to draw your attention to a large isolator room located near the center of this image. The isolator room is utilized to handle the solid drug linker, the high-potent material that requires containment. You notice this individual room has a single positive plus mark indicating positive pressure. The surrounding airlocks and rooms have either 2 or 3+ mark. This indicates these individual airlocks and rooms have a higher relative positive pressure compared to the isolator room. In practice, what that means is that the air in the isolator room does not enter these other airlocks and does not exit the facility. You satisfy the requirements for the high-potent containment while at the same time meeting the GMP needs, and this is a unique requirement for ADC manufacturing. There are other considerations as well. Let's talk about floor design. A CMO may elect to have the low point be in the processing room, in the center of the processing room in the event of a leak. If that leak were to occur, you have the containment aspect of that leak staying at the low point in the room. Additionally, you would not expect to see drains, again, from a containment perspective. Personnel safety is paramount with ADC manufacturing, as some of these high-potent chemicals do present significant hazards. You'd expect operators to utilize powered air purifying respirators and other significant PPE. From a facility design perspective, you must plan this PPE, and you may include additional airlocks for gowning and PPE. And additionally, you want a spacious floor plan, as this PPE can be cumbersome. Finally, let's speak to unidirectional flow. You want your materials, your personnel, and equipment entering at one location in the facility and exiting at a different and separate location further in the facility. And again, this is from a contamination and containment control perspective. Antibody-drug conjugate manufacturing does require solvents. Specifically, the majority of small molecule drug linkers are not soluble in water. Consequently, in the conjugation process, you must solubilize these drug linkers in a solvent such as DMSO. Additionally, you must incorporate these solvents for cleaning and decontamination of that equipment for ADC manufacturing. From a facility design perspective, the facility must be able to handle the flammables and combustibles, as well as it needs to be scalable for what the batch size may be. You'll incorporate engineering controls within the facility for handling these combustibles. ADC, as Jyoti mentioned, is an evolving technology, and a CMO must plan for that future technology. We've already seen some changes when it comes to ADC manufacturing. Approximately 10 years ago, when we first got into the ADC business, we saw just about none of the processes coming into our manufacturing facility requiring advanced purification, including chromatography. Today, we see approximately 80% of new inquiries utilizing chromatography. From a facility design perspective, the CMO must be able to plan for these upcoming future technology changes. Additionally, scale variability is important. Today, for the most part, we're seeing clinical scale sizes, which is typically between the 100 gram to 600 gram of MAB input. But a CMO must plan for the commercial scale size that we would anticipate to see in the future. This could be as large as 3 or even 5 kilograms plus. You need to handle the flammables and the volumes required to be successful in manufacturing. Demand is important as well. We expect with this robust pipeline to require additional manufacturing batches of ADCs, and a CMO must plan to either have additional processing rooms or have the ability to quickly expand. Single use will be a big conversation in this discussion today as compared to fixed equipment, and a CMO must consider what works in their facility. Let's talk about some of the fixed system and fixed equipment required for ADC manufacturing. First is a drug isolator. This is a location where you can safely weigh the high-potent drug linker. They can be single-use or multi-use, and typically, for a containment perspective, they have negative air pressure compared to the room. Second is a fill isolator. This is a location that provides environmental control for the final fill operation. Specifically, you filter this material into its final bags and or bottles, and you want a grade A space and a controlled fill isolator to do this operation. Facility WFI, water for injection, is paramount for ADC manufacturing as well. It's utilized in buffer and solution preparation, equipment cleaning, facility cleaning, and really it establishes your microbial control strategy from the beginning, as buffers that you prepare using WFI are used throughout the process for ADC manufacturing is important for biologics. Autoclave and clean steam are a little bit more optional, but provide a CMO flexibility. Essentially, a CMO can prepare custom assemblies to meet different processing needs, and then autoclave those assemblies to ensure microbial control. Clean steam is utilized to support that autoclave, but can also be considered for clean-in-place opportunities. The last two items mentioned here, parts washer and facility air supply, are also optional. Parts washer allows a manufacturing facility to quickly wash equipment, and a facility air supply is utilized for integrity testing. So let's focus a little bit more on the fixed versus single-use equipment discussion. So a CMO facility must make decisions when it comes to incorporating fixed equipment. Fixed equipment requires a very large footprint within that facility, and a CMO is expected to have multiple processes that require flexibility. In addition, as we spoke to before, there are new technologies that we need to incorporate in ADC manufacturing. Consequently, a CMO may select to not incorporate a large amount of fixed equipment and instead may elect to utilize portable or disposable single-use equipment. On this next slide here, we actually have an image of the inside of our ADC primary manufacturing facility. This is a primary processing room. One item that I want to note is that for the most part, this facility is largely empty. You don't see a large amount of fixed equipment. But on the left, you do see a portable reactor. And again, you can set up a process to meet the needs by using this portable equipment. On the far wall to the left, you also see some of the system requirements utilized for ADC manufacturing, WFI drops, air supply, and engineering controls for handling solvents. So let's talk about opportunities for single use when it comes to ADC manufacturing. The first that we'll speak to is manufacturing materials. This includes tubing and filtration assemblies, buffer and solution bags, process fittings, and aseptic connectors. This is well accepted in the biopharmaceutical manufacturing world and can be utilized in ADC manufacturing. It's an evolved technology, but it provides microbial control and flexibility for a CMO. Next, let's talk about some of the more complex opportunities. First is TFF and ultrafiltration systems. These systems have the opportunity for disposable membranes and disposable flow paths. Both of these can be single-use in a process. This is a widely accepted technology and currently being utilized in ADC manufacturing. Next is chromatography. As we mentioned, this is relatively new in ADC manufacturing, but has been successfully incorporated from a single-use perspective as well. What you have here is single-use flow paths, and slightly more complicated here is single-use columns as well. There's a lot of advantages for our clients to utilize single-use columns, especially dependent on the phase in which that client is currently working. Finally, we'll speak to conjugation reactors. So this is the most emerging of the technology that we have listed here today. What we're talking about here are single-use reactors in the ADC manufacturing process. These are used for the conjugation of the mAb and drug linker, typically. These reactors can be jacketed, and they can have built-in magnetic mixers. There's a lot of opportunities here. So in summation, there is an opportunity to have an entirely single-use system for ADC manufacturing. It's feasible, flexible, and we are currently incorporating that at our CMO today. There are significant advantages and some perceived challenges that come along with utilizing single-use equipment. When it comes to the number one advantage, it's minimizing process carryover from a quality perspective. Essentially, this can be between different processes or even different lots of the same process, but when utilizing single-use equipment, you use it one time, you dispose of it, and there's no risk for carryover. Second is enhanced microbial control. The majority of these single-use items can come pre-irradiated, can be autoclaved, or come in sterile solutions. And you start your process with quality by design with a sterile process. Transportability is very important as well. What you're looking at here is essentially if you're using single-use materials, you can easily transfer that to a second facility for continued manufacturing. It's a quick process from a project perspective. Fourth, we'll mention removing cleaning validation requirements. And from a project management perspective, this can be a significant and timely exercise for our clients. And when you're using single-use equipment, you remove this requirement. Additionally, single-use equipments oftentimes have shorter lead time compared to fixed equipment. There are perceived challenges, and some of these are legitimate challenges that must be overcome when utilizing single-use systems. The first and the greatest concern that we receive from our customers is the potential for leaks. We understand as a CMO that the input on these ADC manufacturing events, the mAb and drug linker, have high investment costs for our clients and additionally have high lead times. We are manufacturing biologic products here with ADCs as well, and a leak could lead to lost material, or in the case of biologic, microbial contamination that leads to a lost batch. When it comes to mitigating leaks, there are some advanced options here. Film technology is advancing and being developed to meet the needs here to prevent leaks for these single-use equipment. Additionally, the manufacturer of these single-use items can provide leak testing at the manufacturing location, or the CMO can even perform leak testing or integrity testing once they receive these different single-use equipments at their location. Training is also paramount when it comes to utilizing single-use equipment. Specifically, a CMO should have operating procedures for handling these different single-use equipment, how to install it, how to ensure that a puncture won't happen, and removing any sharps or other objects that may cause that to happen. We as a CMO have a history of success in not having leaks in our process. Next are process limitations. You must consider temperature, mixing, and solvent compatibility in ADC manufacturing. As far as temperature, we see in practice the highest temperatures around 37 degrees Celsius, and these different bags and assemblies are adequate at these temperatures. Next is mixing. Mixing is typically a critical process parameter in these different manufacturing events, and what you find is that there are mixing studies to show exactly how long you need to mix utilizing these different reactors. Third is solvent capability. We do understand solvents are introduced in this process. For the most part, these solvents are compatible with the different material construction, and additionally in ADC manufacturing, it's diluted by the time you're in the single-use systems. Scale limitations are a concern for our customers as well. Essentially, you need a process that you can meet the needs at the highest commercial scale while providing demonstration small-scale models that can easily transfer up to the large scale. So our clients are concerned about both of those. Do we have demonstration units, and can we meet the commercial need? And the answer to that question is yes. The manufacturers are understanding the needs for our clients, and they're providing on both ends of the spectrum today. Like I mentioned earlier, we are incorporating this process today with demonstration leading to GMP at our CMO. Finally is leachable and extractables. In many ways, this is the least of concern for our customers. The material constructions are typically in acceptance with the ADC manufacturing, and you may need to do some analytical studies, but that's a go-do activity. So here's an example of some of the single-use systems. On the left is a TFF system, and the white box there on the left image is an example of that, where that disposable flow path would be installed. And on the right are some holders for the single-use bags and mixers. So we talked quite extensively about building a facility, but as Dr. Gupta mentioned earlier, having a robust environmental monitoring program is critical for success when utilizing that facility. When it comes to designing an EM program, a CMO must consider from a risk-based approach where and how often to sample different locations. It also must consider the operations that are being performed in that manufacturing facility. A CMO may elect to include dynamic monitoring as well, essentially doing environmental sampling at the time of open process steps. ADC manufacturing in general has more open process steps as compared to other biologics such as MAB, and this dynamic monitoring is important. The next two pieces that we'll speak to are not necessarily unique to ADC manufacturing, but are critical for success. That's establishing your gowning and the operating procedures that go along with the gowning, and then training on both aseptic technique when working in the facility, as well as training on that gowning. There is an opportunity to perform gowning qualification as an analytical method of showing compliance when it comes to gowning and performing operations, specifically operators gown. They simulate an event in the clean room, and then they touch media plates, which are then cultured to show the success of that particular event when it comes to microbial control. Next, we're going to move into process and analytical development strategies. And there's really two buckets here. The first bucket is specific to that ADC when it comes to its function and toxicological use. The second is microbial control. We're going to focus primarily on the microbial control and building that microbiological quality into the process. But I'm going to speak at a very high level of the individual needs from application perspective. You need to consider the MAB, the drug linker, and the ADC. For the MAB, it's the binding assay, binding to those cells. For the drug linker, it's the presence of potential conjugatable impurities, or are there impurities in your solid drug linker that further downstream may conjugate to your ADC? These are things that you need to evaluate. Finally, when your specifications for your specific ADC, you need to consider your drug-to-antibody ratio, as well as your residual drug linker, which both contribute to a toxicological perspective, as well as your residual solvent, your ID, and those complex bioassays that Dr. Gupta was referring to earlier. Let's focus on microbial control. For microbial control, you need to understand your contributors for endotoxin and bioburden. That includes raw material, equipment, and personnel. You also need to understand the facilitators for those. That's process conditions, or temperature, and time. A CMO needs to have a comprehensive strategy for microbial control, and it begins at an early phase and must expand into the commercial phase. Again, this goes back to building that quality into your process. At an early stage, a CMO should be performing a quantitative endotoxin contribution assessment. Essentially, you look at all of your raw materials, you look at the specifications for those raw materials, you do quantitative assessments to say, if we were to have all of the endotoxin at the high end of our process, what will we see in our final product? It's a paper exercise that you can do at the beginning to ensure success. As you move into GMP for your first GMP batches, a CMO should be collecting in-process bioburden and endotoxin results through different processes. Additionally, they need to understand the process endotoxin clearance opportunities. As we mentioned earlier in this talk today, chromatography has been included into this manufacturing process for ADCs, and in some cases that can provide endotoxin clearance. This should be documented early in the development phase. Additionally, you should utilize facility and batch data from previous ADC manufacturing events and combine that with the information that you're gaining as you do your first GMP batches as you move towards commercial to understand what alert and action levels should be in place for your process. That leads to your late-stage commercial microbial control strategy. As you're walking towards commercial, you can add in-process filtrations at your high-risk locations where you're seeing the highest input of microbes. You can perform hold-time studies, both looking at your chemistry and your microbial aspects to understand what if, in case you need to have hold times in your process. Third is solution stability studies. Again, this goes back to demand for a CMO. If you establish 30-day or 60-day buffer stability in your buffers, you can perform campaign batching, and that's important for our clients as a CMO for producing ADC product. And finally, a CMO should consider incorporating closed final fill processes, as that provides the greatest level of control from microbial contributions. We talked quite extensively about single use, but in the case that a CMO elects to utilize fixed equipment or multi-use equipment, they need to establish a cleaning strategy. For ADCs, you must evaluate your MAB and your MAB degradants, your drug linker and your drug linker degradants, and the ADC and those ADC degradants. So how do you do that? You need to have a quantitative analytical approach. For your drug linker components, that's looking at a mass spec method, and for protein, you're looking at UV. One method of doing this is to perform coupon studies. I won't go into too much detail, but essentially you get a coupon of your material of construction. You analytically prove using those techniques that you developed analytically before that I mentioned, mass spec and UV, to show that you can recover your material from coupons immediately after you swap your material onto those coupons. And then you affix drug linker or your other components to your coupon. You allow time, and then you utilize a cleaning method. With your analytics, you can show that you can clear that material. Finally, from the degradants perspective, you can essentially perform stability studies of your individual components in that cleaning solution. If it's stable, you don't have much of a concern for showing clearance of those degradants. All of this feeds into the maximum allowable carryover when it comes to an overall cleaning strategy. And you need to evaluate the potential carryover for your process as well as your specific ADC for what a concentrated dose and how that could impact if it was still in the cleaning after you perform the cleaning event. This in many ways is product specific. All of these different items lead into the regulatory requirements, and Dr. Gupta talked extensively about many of these. Process development and characterization can be done by our clients or can be done with our clients working with the CMO to identify these critical process steps and process parameters, as well as range finding and characterization. It feeds into PPQ qualification, process performance qualification. You need to have completed equipment and method validation, prepare a PPQ protocol, execute 3 batches and release, and finalize that PPQ report for filing. As Dr. Gupta mentioned, continued process verification is critical here. Annual analytical performance assessment will show that your process is under control. In summary, ADCs are a new class of molecule. There's not any ADC-specific regulatory requirements today, but at the same time, there's a robust ADC pipeline driving demand. In many ways, this means that the CMO must act both as a partner in science as well as a partner in quality. This is founded in many of the items that we discussed today. Facility design, environmental monitoring, process development, and regulatory strategies all contribute to success in manufacturing ADC and must be considered by the CMO. Finally, this is an evolving technology. A CMO must be ready for the future. That's incorporation of these single-use opportunities and what other changes may come in the future for ADC when it comes to purification strategies. On that note, I want to thank everybody for listening to our talk today, and I'm going to pass this back to Afshal.
Thank you, Josh, and thank you, Jyoti. At this point, I would like to begin the question and answer session, and the first question we have is, for implementing QBD for biologics, is it required to evaluate or determine critical process parameters at the full manufacturing scale?
Thanks, Afshal. This is Rajesh, and I will attempt to answer this question. Is that this question is often asked, and it is a very relevant question because during development you don't have a full manufacturing scale, and I have not seen any of these critical process parameters being defined based on the full scale. You have to go on a scale down, and if you are in a commercial manufacturer already working on some product which is already done, or during development you take a relevant scale which you can scientifically justify, and then later on when you go with the commercial manufacturer, as I mentioned, and I think Josh also highlighted or emphasized that the continuous monitoring of the process and verification, then you verify those critical process parameters with the commercial scale, which were initially identified with on a lower scale or scale down process. So I think in the practical terms, I would say that you have to work on a scale down manufacturing process, and just you need to justify that how it is scientifically justified that this process ultimately will represent the full commercial manufacturing scale.
Thank you, Rajesh. I think we have time for one more question. I've got a question here for, I think, Jyoti and Josh. Can you or do you use ADC facilities for non-potent conjugation?
Yeah, I'm going to have Mark respond to that.
Certainly. Yeah, utilizing the commercial ADC facility for executing or processing non-potent conjugations is certainly something that we would allow from a quality standpoint, given that you've established the same protection of product carryover and shown that you can effectively clean and remove any potent conjugated products from the facility before you would embark on manufacturing a non-potent conjugation product. So really the same rules apply. You've got to have a very strong and robust cleaning program, and then the analytical methodology to show that you've cleaned the equipment and facility appropriately. But the space is designed to execute conjugation reactions. If you're fortunate enough, like we are, to have a separate facility where we could actually execute non-potent conjugations, that obviously becomes an option as well. But if you don't, I don't see that any quality objections to doing that, given the requirements that you have to show before doing it.
Yeah, if I could add a point, as a CMO, we do provide the options to our customers. Sometimes it's customers' decision whether they want to share the ADC facility for a non-ADC type of product. So it's ultimately their decision.
Thank you, Mark, and thank you, Jyoti. Okay, unfortunately, that is all the time we have. Any questions that have not been addressed here live will be answered via email. I would like to take this opportunity to thank our presenters, Dr. Rajesh Gupta, Dr. Joji Swami, Josh Morris, as well as Mark Cooley, for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com/webinars. Thank you, everyone.
Thank you. Bye.
Thank you.